Okay, guys, so now that we've looked at DNA replication and also the fact that DNA is used as a code for proteins, let's check out the errors that can occur when DNA is being replicated and how that would affect the code that is coding for the proteins, the amino acids that we have talked about. There are a couple of different kinds of mutations that can occur, errors within the DNA code, and we kind of in general could split them up into two major categories. One of them would be a spontaneous mutation. So spontaneous, you might want to think of it like there was a mistake made, okay? A spontaneous error occurred in perhaps the replication process that resulted in the incorrect nucleotide being placed, being paired with another. Versus inducible, we're not talking about inducible operons now, inducible mutations, if there's an induced mutation, that would indicate that there is some specific chemical that causes the mutations, okay? Some external cause of mutation, okay? So what I'm going to look at first with you guys is more so, I guess, the spontaneous mutations. So let's say we have replication going on. We have our, I keep doing it, but you might think I'm crazy, but 5' to 3', right, that come apart. And then we start to read and place those nucleotides, those base pairs. What if during that process where DNA polymerase is placing those base pairs, there's a mistake, right? Right there during the process. What can occur is that immediately the correction can be made. Now, the analogy that I make when I go over this is typing along, right? I'm a horrible person at a keyboard. I'm always staring at the keyboard instead of the screen like I should be, but I'll be typing along and I'll look up as I'm, try to look up as I'm going, and I notice that I misspell a word. And if autocorrect isn't on, right, or sometimes that doesn't correct it the right way, right, but what I'll do is I'll notice that it's actually made an error, and I'll just backspace and put in the right letters, right? And that's more or less what DNA proofreading is. This situation where the DNA polymerase is placing nucleotides, and then it almost can backspace and place the correct nucleotide for an error that's made. Now, DNA repair, a specific type of this called mismatch repair, is a situation where the replication process has completed, and then immediately following replication, it's noted that there's a mistake in the code, okay? So my analogy for this would be, I'm typing along the way I do, because I'm not very good at it, and I'm typing along and I look up and I notice that, okay, I've written half a line or a couple lines, and I see, or a whole paragraph, and I see that 3 or 4 mistakes are made. Well, I don't have to backspace. What I can do is I can specifically go back to those places where I have errors and correct them. This is a mismatch repair. And this would require a slightly different set of enzymes, but again, it's a way to deal with mutations like this, spontaneous mutations. One is DNA proofreading, it's like backspacing. Another is mismatch repair. Finally, we have another kind which is called nucleotide excision repair. Now this can be, this is an ongoing process where the DNA in cells are constantly being rechecked and rechecked and rechecked. And then the detection, removal, and repair of damaged DNA is being done. An example of that might be like this example here, where there's two thymines are bound together side by side. This shouldn't be. This happens with UV light, causes mutation to DNA, maybe in your skin cells or something. And nucleotide excision repair can be checking for these all the time. Locate the errors and repair those errors, okay? So this analogy might be, you didn't backspace, you didn't see it after you wrote the paragraph, but a week later you're kind of proofreading this. I say proofreading, I should say you're checking this essay that you wrote, right? Or you give it to a friend and they read it and they see immediately there's a mistake there, right? That would be more like a nucleotide excision repair, okay, where you would do something like that. Something that's worth mentioning is that viruses lack DNA repair mechanisms. They actually have to hijack other cells, and in so doing, a lot of times these viruses churn out genetic material and package the genetic material before these errors can be corrected, okay? Before the natural host mutation repair mechanisms can take effect, the viruses have mutations, and this can sometimes be beneficial, but oftentimes it would be detrimental to a cell. To make just a random mistake would most likely be a detrimental problem when it comes to making that protein. All right, so let's talk about what these mutations can do. I've already mentioned spontaneous versus induced mutations, right? Spontaneous occur just kind of naturally as mistakes are made, versus induced mutations where they might come from a specific chemical that's in an environment or something. But let's talk about point mutations, okay? Point mutation is a single nucleotide change. A single nucleotide is in error in the original DNA code. Now, there are two types of point mutations. One would be a base pair substitution, where in the replication process, the wrong nucleotide is placed, right? Or there could be a base pair deletion or insertion. So in this case, there's no nucleotide placed at all, or there's one completely removed from the sequence, okay? So two different basic types of point mutation. One where we have the same number of nucleotides, we just have one that's in error in place of another, right? Substitute, right? That kind of makes sense. Something's put in place of another, right? And then the other situation where we have an entirely nucleotide is entirely missing, or an entirely new added nucleotide in the sequence. Now, there's lots of different kinds of base pair substitutions. This one type of point mutation called a base pair substitution can occur and have lots of different kinds of effects, okay? So let's consider this particular set of DNA. We have a wild type DNA. So that would indicate this is in the wild. We find these naturally. Think of it that way, okay? So this is a normal sequence right here we have at the very top, okay? And this normal sequence right here would go on to give us A, G, C, G, U, A. And if we go throughout this entire sequence here and look at our chart that we learned how to use before, we'd find that A, G, C gives us serine. G, U, A gives us valine, okay, and so on. So we've got these four different amino acids, and this is our protein. What if we have a point mutation where we have a substitution for this A right here? So in the replication process, at some point long ago, right, or previous to this point, an A was not placed during the replication process, but a T was. So here we have our error, okay? This is a substitution because there's a T substituted for an A, right? Now when we look at our chart, we're going to have A, G, C, G, U, U. Not G, U, A, but G, U, U. Now, if you look at your chart and look to see what does G, U, U give us, well, lo and behold, it gives us exactly the same valine. No real change to the protein has taken place. If a mistake like this is made, there would be no change to the function of that particular resulting product. Okay? So let's talk about some of the different labels we could give these. Okay? One would be a silent mutation. This would be an example of a silent mutation because the substitution has no effect on the function. Now, sometimes you'll find that there are silent mutations even if we have a different amino acid placed there. If they're enough alike, right? If they are both negative in charge, or if they're both non-charged, right? Then they might result in the same shape protein. So it may be, again, silent, okay? Now, this is also a missense mutation in that there is still this codon, this sequence GUU, is still coding for something, right? So it's a missense mutation and it's a silent mutation. So we could say that this change, let's say I gave you that in a question in the test, right? I said, okay, we have a sequence and an amino acid or a nucleotide is changed to a different one, but it has no effect in the function of that protein that results. You'd immediately know many things. You know that it's a point mutation because it's a single nucleotide. You know that it's a substitution because I said that one was in place of the other, right? You know that it's a silent mutation because the protein apparently still works, so it must have made an amino acid in that location, missense. So in that case, it's all four. It's a point mutation, that's a substitution, that's a missense mutation, and it's silent because it didn't affect the function. Okay. All right. Let's talk about this this one down here. Let's talk about a nonsense mutation. Here we have a substitute. So it is a base pair substitution. Okay. So here at the bottom we have the same original sequence, but what got changed is the C at the very end. Okay. And that C at the very end was changed to a G. So there it is. There's the mistake. It's a single nucleotide, so it's a point mutation. It's substituted for, so it's a substitution. But when we look at our chart to see how this will change things, our UAC, which coded for tyrosine, is now UAG in our codons, right, in our RNA sequence. If you look up UAG, go and find your chart and check it out, you'll find that that is a stop codon. That's a termination. There would be no more amino acids strung together for this protein. Now, the fact that this is no longer an amino acid being placed in this location, it's just a punctuation mark. Think of it that way, right? A stop codon is a punctuation for the end of a sentence, let's say, right? At the end of this reading and placing of amino acids. Do you think that protein is going to act the same if an entire amino acid, and remember, it could be that there's a lot of amino acids that follow this. A nonsense mutation is a substitution that results in a stop codon. So here we have it, a nonsense mutation. A codon becomes a stop codon. Usually, and it makes sense, right, that if you just chop a protein off like this, well, it's probably not going to function the same. Okay? So it would probably not be a silent mutation, because you'd probably lose the function of that protein entirely. Lethal. Now, in this case, you think lethal, and it might be like the death of something, right? But lethal in this case would be there is an effect. There is a detrimental effect in the fact that a changed amino acid or a lost set of amino acids drastically changes the effects of that protein function, how that protein functions. Okay? So, in this particular case, we had a nonsense mutation, and most likely it would be a lethal mutation as well, because these cases where we just chop proteins off like that by putting a punctuation mark in the middle would really affect function. So let me give you another example here. So here we have one you all know about, okay? Sickle cell gene. Here we have a natural wild type hemoglobin DNA. So this is making hemoglobin for carrying our oxygen. We have a natural CTT going on, which would code for a GAA in a natural transcript, and we'd have a glutamate, this is called. But you basically have a normal protein for carrying oxygen. In the case of sickle cell, the gene has an A in the place of the T. It's taking its place, so it's a single nucleotide, point mutation. It's taking its place, so it's a substitution. It's still coding for something. Check it out, right? There's still, if you go to your chart, you'll find that it still codes for an amino acid. So it is a missense, not a nonsense mutation. If it was a stop codon, it would be nonsense, right? Wouldn't make any sense because the protein probably wouldn't function. But in this case, it's a missense. The wrong amino acid is now placed in that location in our protein. Now the question is, does the hemoglobin protein for someone who has sickle cell function normally? Does it have an effect? Does it change things? And the answer would be absolutely yes, it changes things. These proteins clump together differently. They change the shape of the red blood cells. They are definitely a lethal mutation as well. Now, this is not necessarily a death sentence, right? I mean, it can be pretty serious, sickle cell, and it can shorten someone's life for sure. But it's not, and so it would still be called lethal because it's affecting the function of the protein product. Okay. All right. So what I usually talk about at this point is I have this kind of corny sentence that I put together. Okay, and they're all three-letter words. So you might think of them like triplets of code, right? And then ultimately the codons that are in the RNA. Okay, so here we have a mashed together sentence, all right? And it starts with a capital letter. That might be your start point, right? And then it has a punctuation mark at the end. That would be your stop point. Think about maybe start codons and stop codons. Now, we need to read these triplets in the correct reading frame. We need to start in the right place reading them, right? Otherwise, it makes no sense at all. So if I split up the letters like this, there's nothing there that makes much sense, right? If I split it up like this, the reading frame has been shifted over one letter. Or think of it like about DNA, one nucleotide. It again makes no sense. The product isn't going to function for us, right? If we split it up properly in the correct reading frame, so here we have a sentence. It's kind of a silly sentence, but at least it has sensible words, right, that are listed together here. All right, now, if there's a deletion or an insertion, we're not talking substitution now. We're saying we're going to take a letter straight out of this. What if that error occurs? What if there is a point mutation where there's a nucleotide completely removed or completely added to the sequence? Like here, I have a deletion. I've taken the U, that particular letter, completely out, this is going, if we keep with the same reading frame, so I got the first letter, think of it like our first amino acid, that has function, that makes sense, but then look at all of the words after that. This is how important reading frame is. If we have a base pair deletion, even if these code for amino acids, these nucleotides, pretend like they're the nucleotides now, the chances that this protein product is going to function are slim. So a deletion of a single nucleotide will most likely be lethal, detrimentally affect the function of whatever protein it produces. Now, let's say we add a letter, right? So I stuck an H in here, right? In between the S and the U in sun. And we still go in threes. Well, again, this makes no sense. So, most likely a lethal effect. So when it comes to deletions and insertions, if the point mutations are deletions or insertions, we're most likely going to have a lethal effect. Protein will not function the same. What about substitutions? Well, I could have a substitution, let's say here where the M is in man, right? And it has a different letter there now. It's a substitution. One's there in the place of another. Well, it doesn't totally throw off the sentence. It is kind of strange, right? So now the man is a fan, right? Okay, so the sentence still makes sense. It's a mistake though. So we say it's a missense, right? It's producing perhaps an amino acid, but that amino acid is not quite the same. Whether this is lethal or not, well, I can make sense of that sentence. I'd say it's not lethal. It's more or less a silent change. Okay. Now, if I change this to another letter, like a V, okay, I know a lot of fans that wear hats. Okay, I know a lot of baseball fans that wear hats. So this kind of makes sense, this first sentence. But now, if we change that letter to a V, well, I don't know any Vans that wear hats, okay? So this would change the meaning. It would change the function of the sentence at its heart, right? So this would be more like maybe a lethal mutation, where it's really changing the meaning of the final product. All right, so this frameshift idea, this idea that if you completely lose or add nucleotides, you would change the reading frame, is really important. Frameshift mutations are very detrimental and usually lethal in the fact that the protein products will not function the same. All right? Okay, so let's call it quits there, guys. I'll see you guys next time. We'll talk about genetic transfer, a pretty crazy thing that bacteria can do.